Good morning, and thanks to OIS for having me here. I really appreciate it. And I'm going to be talking about intermediate AMD and our oral drug to prevent patients with intermediate and who have good vision from progressing to significant vision loss. There are 18 million Americans living with intermediate AMD today, and they have no FDA-approved pharmaceutical treatment available to them. Oral small molecule Xiflam is our drug, and it targets the NLRP3 inflammasome. This is to inhibit recycling inflammation. We have executed an option to acquire a deal with Bausch & Lomb, which drives significant investor return. And we are led by a seasoned team with demonstrated success in approving ophthalmic drugs. So what is the ideal drug for intermediate AMD? If you look at the right, you'll see it avoids intraocular injections, treats both eyes simultaneously, penetrates the blood-retinal barrier, demonstrates good clinical safety and is well tolerated, and preserves structure, function, and disease progression. It's manufacturable to global regulatory guidelines, and it supports economics where health equity is taken into account, which means it's affordable, and also in this particular case, increases the provider base to include general ophthalmologists and optometrists. Xiflam's chemistry enables penetration through the blood-retinal barrier and delivers effective concentrations, as you can see through the figure below. And greater than 2,000 subjects have received Xiflam at different dosages in chronic dosing. So we can safely administer this drug at extremely lower levels than what it's been dosed previously. So it's got a pretty good therapeutic index. Our drug target is the pathologically open connexin 43 hemichannels. Now, these hemichannels are continuously formed in the cells of most organs. They have one function in life, and that is to stay closed till they dock with another connexin 43 on an adjoining cell, and they both open to form the gap junction. In specific disease states, they open prematurely. They signal the ATP gets out into the extracellular space, and they signal the inflammasome. And we have perpetuated inflammation. In this particular model, ischemia and leaking blood vessels secondary to inflammation are integral to retinal vascular disease. In this experiment, we raised the intraocular pressure to 120 millimeters and get the retinal ischemia, and then for 60 minutes later, we reperfuse just by simply letting the pressure come back. You can see there's significant overexpression of connexin 43, but the most important part of this slide is that we reduce leakage by 86% between the placebo and the treated eyes. In this study, where we do a phototoxic model of retinal degeneration, we put a very bright light above the animals, and you can see the retina in the treated eyes is fully functional at 3 months, whereas the placebo retina has lost both structure and function. Our target for the clinical is persistent choroidal hypertransmission defects. This has been published in the American Journal, and this is the results of a seminal natural history study done over 5 years out of Bascom Palmer Eye Institute as a novel biomarker for this particular condition. So the patient entry criteria are enriched due to this study. Central drusen volume of equal to or greater than 0.2 millimeters cubed, and no evidence of persistent HDDs. If we randomize 100 eyes into each group, this will ensure an adequate number that will develop HDDs within 1 year. And this is the clinical study design. You can see the enrollment criteria on the left. 300 patients will be randomized into 2 doses of Xiflam, and a placebo will be the control. The primary efficacy endpoint at 24 months is the formation and growth rate of persistent hypertransmission defects from baseline. And these will be plotted as a slope through all time points to measure the rate of progression between the treated and the control groups. We will do an interim analysis at 12 months and then see what the data looks like and then meet with the FDA. If we're successful, Our regulatory milestone would be to request FDA breakthrough designation and also something called CNPV, which is a new voucher that is now being offered by the FDA if you meet the criteria. And based on what I've seen from the guidance, we will meet that criteria, which means we can submit the NDA with the 12-month data and get a very, very quickly reviewed study. All that we have to do prior to submitting the data to the FDA is actually submitting the CMC ahead of time in order to get that voucher. If this all goes according to plan, which we expect it to, we should get a conditional approval around 2028. If not, and we have to go for the full 24 months, that'll take us out to 2029. We have an industry-leading partner, Bausch & Lomb, and we have a comprehensive IP portfolio with 24 patents pending and 9 patents granted, all the way out through 2040 and beyond. And with Bausch & Lomb along with us, we'll have geographic coverage for a drug which we believe will be a paradigm shift in the way we treat patients who are developing macular degeneration. Thank you.